from the original beginning to the original end, which again will become important later on. So what we can then do is we can take the DNA, we can make many, many copies, we can take an aliquot and then put it again into one of those sequencing machines. However, there is still a problem, and that is the problem, like I explained to you before, that there's just such a tiny proportion of the DNA that is actually pathogen DNA that we can study. So if we take some random skeleton from the medieval time, what we would find is that we have about 4% of the DNA to be human DNA that we get out of the fossil. Only about 0.005% is mitochondrial DNA, and a much lower fraction is then actually the pathogen DNA. Which means that if we want to study 1,000 base pairs of the pathogen, we need about 40 million DNA sequences, which is possible. But if you then were to get a concatenated piece of, for example, 1,000 base pairs, we need about 40 billion DNA sequences, which is again possible. It would just be 40 high-seq runs, which is affordable, costs maybe €500,000 or something like that, but certainly not for a Mickey Mouse lab that I have. So that is really out of the question. So that is nothing that we could really work with. So we couldn't use shotgun sequencing. We couldn't do that. So we have to find some other way how we can get out that DNA, but not with PCR, which is so biased. And what we then used is kind of a fishing approach, where we fish out DNA. So it's called targeted DNA enrichment. And there are different strategies that one can use, and two I want to show you today. One of the first strategies we use to get out this PCP plasmid. Like I told you, the PCP plasmid had many copies, so there's a lot of it in the bone, so it's easier to actually look at it. So what we then did is we took skeletons from a site in London, so it's called East Smithfield, and that was a field which was purchased in the middle of the 14th century during the Black Death, so we know that from historical records, just to dispose the dead people. Because there had so many dead people in the streets, they had to somehow bury them. They don't wanted to burn them because they were Christians, so they had to somehow get big fields and basically dump the bodies in there. And that was only used for 2 years, and there were approximately 10,000 people buried on that field. And that field was actually rediscovered in the 80s when they built some apartment complexes there, and they were digging, and suddenly there were a lot of dead people. So they called the archaeologists. The archaeologists came in and they took out all those skeletons, at least the ones that were just below this apartment complex, about 500 skeletons. And those we could actually study a few years ago. So we took about 50 samples from those 500 skeletons, and in 20 of them we actually found with PCR that pestis DNA is present. So we knew, okay, there seems to be something there. Now we just have to show it's real, it's ancient, and there's a lot of it there. So what we then did is we extracted the DNA, we immortalized it, turned it into one of those DNA sequencing libraries, made many amplifications, so we have many copies. So we have a high amount, we can produce micrograms of DNA, and that DNA we can then use for this DNA capture. And the protocol we used was developed by a PhD student in the lab back in Leipzig, and there was a protocol where you use long-range PCR products as a bait. Because if you think about how do you go fishing, you usually need a bait, and somehow a pond where you want to pull the fish out. And the bait here was produced by long-range PCR products, so we took modern pestis DNA, we made long-range PCR products, they were broken into small pieces, then you attach a hook to the bait, and then this hook, which is a streptavidin here, biotin streptavidin, so the biotin here on the bait binds to beads which are coated with streptavidin, and what you then get is a little bait ball. So it's a little ball which is coated with the bait DNA, which is magnetic, which is really handy. So you can basically throw the bait ball into your soup, which is that complex DNA, and then all the fragments which are complementary will bind to the DNA on the bait ball, and because it's magnetic, you just pull it out, and then you have your DNA basically enriched for the target DNA, which is really great. So we put the entire plasmid on those bait balls, and we could then fish it out. And that worked rather well. We not only did it for pestis DNA, we also did it for human DNA, because we wanted to make sure if we get pathogen DNA out of those fossils, we should also get human DNA. Otherwise, something is weird. So we also fished out human mitochondrial DNA, and we used the controls. We used, first of all, a collection of skeletons that were 100 years older than the Black Death. So they should not have Yersinia pestis, because it wasn't present during that time. When we used those control skeletons here, we actually found a lot of human DNA, enough to put together complete mitochondrial genomes. but we found not a single fragment of the PCP, of the Yersinia pestis DNA, which was really great for us to show, okay, it was not just soil bacteria, which are just around all over the place, which of course potentially could be the case. But then if you look at the East Smith field, we found again a lot of mitochondrial DNA, but we also found some 20,000 PCP fragments, actually enough to put together the entire plasmid, which was kind of a good proof of principle for us that there is indeed Yersinia pestis DNA present in those samples. But then again, we have the problem of showing that that DNA is authentic. We have to somehow show that it's not just lab contamination. I told you we used modern PCR products. Maybe they contaminated the lab. Maybe they get somehow into one's clothes when we went into our clean room facilities or something. So we somehow have to show that that DNA is old. And I have actually developed some years ago a method to look at DNA and prove that that DNA is ancient DNA by looking at DNA damage. So I told you the ancient DNA gets basically chemically modified over time because it's old and everything degrades over time. So things just change. That's just how it is in life. And then when you have a DNA fragment like that, which is really old, what you often have is that the end of the DNA is single-stranded. And single-stranded DNA has a very high rate of damage. And usually the damage that you see is deamination of cytosine. So what you get is called miscoding lesions. So when you look at the DNA, you, for example, see instead of Cs, you see Ts. Because cytosine was deaminated. Deaminated cytosine is uracil. And uracil gets read as thymine by the polymerase. So you get a C to T change. And when you look at the damage plots, so you walk along the fragment and you look where you see damage, you can actually see that this damage is highest at the beginning of the fragment because it's single-stranded. And then when you walk along the fragment, that damage gets lower. And then at the end, it actually goes up again. But what you see going up is not C to T, it's actually G to A, which is not a damage we know of, and it's a really weird pattern. But that has something to do with how we prepare the DNA for sequencing, because we have a fill-in step. We have one step where we fill in this overhanging ends here. So we actually fill in the overhanging 5' ends here to the kind of 3' strand here gets filled in, and then you get actually the reverse complement. So if you had a C to T occurring on this side of the strand, you actually get a G to A on the other side of the strand. That's exactly the pattern we see. C to T here, G to A there. But what was really surprising when we first observed that is that you see about 40% of all the cytosines to be deaminated at the beginning of the fragment when you have, for example, Neanderthal DNA. But if you use, for example, contamination in the same Neanderthal bone, you see almost nothing. So modern DNA, of course, doesn't show that. Because it's not old, it's not deaminated, it basically hasn't changed over time. And we have done some more studies now, where a student of mine looked actually at bones that span the last 40,000 years. So she looked at bones that are 20 years old, 50, 100, 500, and so forth, up to 50,000-year-old bones. And she looked at the rate of deamination, and you can actually see there's a really nice correlation. So if you have really old things that are usually pretty high deaminated, if you have young things, usually the deamination rate is not so high, so you have less than 20%. And that, of course, gives us now the possibility to say, if DNA has this damage, it should be old. If it doesn't have that, it can be contamination, it can be old, but it should not be old. And that's really handy. You can actually also see, when you look at that, there is one group of bones which look a bit funny here. Those bones are actually very special bones. They're gorilla bones from Africa that we got from a museum, and we found them to be really weird, and we asked them, what about those bones? What happened to them? And they were like, yeah, they're kind of funny. They were actually butchered by people, so they caught those. They actually cooked them. Then they disposed the bones behind the house, and then some year later, anthropologists came along and, you know, do you have some bones of gorillas? And we're like, yeah, we just have a whole pile of them behind the house. And they actually excavated those and then put them in the museum. So they were cooked and excavated and then put in the museum. So as long as you don't have bones like that, usually you're pretty fine. So that's really special. And even those bones that are really heavily treated, they have below 20%. So our criterion right now is usually if it has 20% damage, you can believe it is old, which is quite handy. So if we then look at the bones again from London, when we first look at the human DNA that we got from those 1,000-year-old bones, we see it has about 25% damage. So that is real ancient human DNA. We can also show that that DNA is just from one person. And if we look at the Yersinia pestis DNA from the same specimen, we see about the same rate of damage, so about 25% damaged DNA. So that is old Yersinia pestis DNA. That cannot be just let's say 5 years, 10 years, or 100 years old. That is at least 150 years old. Likely it is more than 500 years old, because if we go back, we say 25% damage, we're somewhere here, so that would be between 500 and 2,000 years, which is kind of perfect. That's exactly where we want to see it, so it should be really old. So from this first study, we could then conclude, first of all, we have authentic human DNA, which is good, so there is DNA in those bones, but what we also have is authentic Yersinia pestis DNA, so there is pestis DNA in those bones. What we could also say then is that Yersinia pestis, so what causes bubonic plague, was actually one of the causative agents of the Black Death. Of course, we cannot exclude there was something else around, but certainly it